Hi, and welcome to the Johns Hopkins Cerebellum Seminars. Today, I'm very happy to introduce to you Mario Prsa. Mario was born in Croatia and immigrated to Canada with his family when he was quite young. He studied engineering at McGill University, where he developed a strong interest in neural control of movements. He then returned to Europe and pursued a PhD under the mentorship of Peter Thier in Tübingen, focusing on cerebellar and brainstem circuits involved in oculomotor control in macaques. He then, as a postdoc researcher in the laboratory of Daniel Huber at the University of Geneva, explored artificial sensorimotor learning, vibrotactile encoding, and sleep-related mechanisms in the mouse cerebral cortex. Since October of 2019, he's been an assistant professor at the University of Freiburg in Switzerland, where his laboratory investigates cortical, spinal, and cerebellar circuits underlying forelimb proprioception. Today he's going to be talking about his work and also particularly the work of one of his graduate students, Irina Scheer, who just defended her PhD. Thank you, Mario.
Okay, thank you very much, Prsa, for this kind introduction and invitation to the seminar series, which, again, has been mentioned before. It's really a fantastic resource for the community. It's a really great pleasure to be able to participate. So today I will present some of our recent work in the lab on climbing fibers in the mouse cerebellum and how their activation by different type of sensory events might or might not influence behavior. And as Reza mentioned, this is largely the work of Irina Scheer, who's the PhD student in my lab. So essentially, I will present the work that she did in her thesis recently. Okay, so I assume everyone here is familiar with this motif of the canonical cerebellar microcircuit. So the fundamental principle here is that the key process, or one of the key processes for the cerebellum to enable efficient motor control, occurs here at the dendritic tree of the Purkinje cell. So here the mousy fiber pathway conveys rich sensory, motor, and contextual signals. And then the second input, the climbing fiber, conditions the signals at the parallel fiber synapses and thereby instructs which of these signals should be mapped and how they should be mapped at the output of the Purkinje cell. So the goal of Irina's project is to try to have a better understanding of this instructive climbing fiber signal and the type of information that it encodes. So the classical understanding is that climbing fiber activity encodes errors during movements, and these errors can then serve as a template for learning, so that by adjusting the efficacy of certain of these parallel fiber synapses through LTD and LTT mechanisms, these movements can be progressively adapted and the errors reduced. Now we know that climbing fibers can be activated by many other sensory stimuli in the absence of movement. So I'm highlighting here data from the Prsa lab, but this has been shown by many studies and even in the very old cerebellar literature. The most effective stimuli seem to be tactile and proprioceptive perturbations of a particular body part, especially when they're unexpected. So in this case, an air puff delivered to the face of the mouse. And we know this because the olive maintains a well-defined somatotopy of the contralateral body and is activated, driven by sensory afferents from the periphery. And in addition to these tactile and proprioceptive inputs, so-called teleceptive stimuli that have no physical contact with the body can also reliably drive olivary neurons. So here, auditory tone or a visual flash. And these are more likely conveyed not from the periphery, but via the mesodiencephalic junction and the superior colliculus. So now this broadens our understanding of the olivocerebellar system, which in addition to errors can also encode heterogeneous sensory saliency signals that can instruct plasticity and learning of many different processes, of any process that might be able to influence behavior. So we also know that in addition to these long-term plasticity effects, and learning that climbing fibers can also trigger an immediate real-time instruction and modify behavior in very short time scales. So this is revealed by these types of observations. So when the inferior olive is lesioned, it doesn't only impair learning, but it also produces an immediate movement deficit. So in this case, cats were trained to perform reaching movements with the forepaw, grab this handle, and during the actual task, they injected cyanic acid in the inferior olive and lesioned the olive, and within minutes, they observe these modifications in the reaching trajectories. Essentially, they observe a cerebellar-like motor disorder. And this is also demonstrated in this study from Tim Ebner's labs. And here, monkeys performed a manual tracking of a visual target using a robotic manipulandum. And when they aligned the simple spike firing rate here in blue and the movement kinematics in green to the occurrence of a complex spike at time 0, they observed that the encoding of these kinematics and the simple spike activity immediately changes. So in this case, the encoding increases as shown by these higher R-squared values. And after the complex spike also produces an immediate significant modification or perturbation of these movement kinematics. So again, this indicates this additional role for online motor control of the climbing fibers. And also, as we heard recently in past seminars and recent publications from the Shadmehr lab, using this very elegant analysis, so the presence of a complex spike pulls the trajectory of the isocad in the direction of that complex spike's preferred direction, and thereby defining an instructive potent vector for each Purkinje cell. So inspired by these findings, we then hypothesized that these so-called saliency signals in rodents can also be informative of the immediately ensuing motor output, not necessarily in the presence or absence of complex spikes, but perhaps in the probability of their occurrence. So for example, we reasoned that the probability of a climbing fiber being activated in response to, say, this auditory tone might be informative about whether the tone cues a movement or not, or perhaps cues a fast versus a slow movement. Or if we take the tactile cue, the question is, are the climbing fibers informative about whether this stimulus perturbs behavior versus trials where the mouse might counteract such a perturbation? And we also ask, does the sensory modality play a role, and also the complexity of the climbing fiber activity in the amount of the encoded information? Okay, so to test this hypothesis, Irina transfected neurons in the medial and the dorsal accessory inferior olive with a calcium indicator GCAMP8, which is then expressed as shown here in the axons and in the axonal terminals, in the Purkinje cell or molecular layer in the contralateral cerebellum, which she then imaged with two-photon microscopy, as shown here, through a cranial window that she implanted over lobules 5, 6, and simplex. Okay, so before going into the data, I will briefly take you through the behavioral task. So Irina trained mice to hold the endpoint of this simple one-dimensional robotic manipulandum. And then at the start of the trial, the motor operated in position mode. So it was actuated to stay at this initial position. And the mouse had to continuously hold this lever. And after 2 seconds of continuous holding, we applied the first sensory stimulus, which was a force. So essentially, the motor switched into current mode and applied a small constant force acting toward the mouse. And then after a short fixed delay, the second stimulus was an auditory tone that acted as a go cue for the mouse now to push this manipulandum forward against this force towards a fixed target. And once the target was reached, then the mouse had to continue holding again the manipulandum during this delay 1 second period before receiving the reward. So just to summarize, we have these 3 successive stimuli: the force, the go cue, and the reward. And also I'm highlighting here 2 waiting periods, which will be relevant when describing the results. So in this period here, the mouse is waiting for force for the child to start, and here the mouse is waiting for reward. In both of these periods, the mouse actively holds this manipulandum, cannot let go, otherwise the child will be aborted, but no stimulation or movement occurs in these periods. So now to test our hypothesis, two climbing fibers that respond to each of these sensory events, are they informative about the behavior that follows these events? We classified trials into different categories. So each of these events was followed by different forms of behavior. So for example, after the force onset, this force either perturbed the limb position, as shown here in one trial, or shown here in the average velocity trace, or mice actually counteracted this force, so they produced an isometric contraction during the holding period, which was sufficient to counteract this force, and we call these adapted trials. So here then we classified these trials to follow the force onset into these two categories. Then after the go cue, the movements could be either cued, as shown here, correctly timed, or there was an absence of movement, or in certain trials, the movement started actually before the go cue. They did not cross a threshold to abort the trial, but they slightly drifted already starting before the go cue. So here we classify the trials in these two categories that follow the go cue, either cued movements or no or uncued movements. So we grouped these two trial types because none of them is actually initiated in response to the go cue. And we also then classified only the cued movements into slow and fast movements, just based on the median peak velocity. For the rewarded trials, we classified them into the short versus long latency trial. So after the reward, there was much lower dimensional behavior, not much variability in the behavior, but we nonetheless looked at whether putative or potential reward-related responses would be informative about short versus long peak latency. Okay, so I will just now first describe the type of responses that we recorded or imaged here. So here we have single mode cells, so fibers that responded just to one of these stimuli. So here we have the force-only fibers, the reward-only fibers, the go cue-only fibers, and the reward-only fibers here. So this has been previously reported in these regions of the mouse cerebellum that climbing fibers can reliably respond to many different sensory stimuli. Now, in addition to the single-mode fibers, we more often actually observe the multimodal fibers. So they respond to any combination to two of these stimuli, but and certain fibers also had a trimodal response where they were activated by all three sensory events. So this means that these climbing fibers have the property of mixed selectivity, as been defined in a recent review article by Kei Tai and co-authors, meaning that they can respond to multiple statistically independent variables. So if you now look at all these categories, it is, well, somewhat surprising that more climbing fibers in these forelimb regions of the cerebellum were actually responsive to this teleceptive auditory go cue and less responsive to the force, this proprioceptive stimulus that is directly delivered to the limb. And actually, the force-only fibers were actually the smallest category. The distribution across the lobule is as one might expect. So the force climbing fibers are predominantly found in lobule 5, the reward fibers in lobule simplex, and there was a uniform distribution for the go cue activity. So I should say here, so these are imaging data that was collected in highly trained mice. So after performing this task over many weeks, up to 40 sessions that the mice performed this task in to collect this data. And now more recently, RENAC has actually collected and analyzed data from naive mice as well, from only the first 5 sessions of performing this task. And it looks like in naive mice, these climbing fiber activations are very different. So this is still preliminary analysis, but what she sees is much fewer go cue responses and much more weaker go cue responses, both in the unimodal and the multimodal fibers. And there's more force-related activity again in both of these single and multimodal responses. What's also striking here is that we do not see any unimodal reward-related activity. All the reward-related activity, which is quite prominent, is only present in these multimodal responses in these naive mice. So then if we then compare the naive and expert mice, we see that the force stimulus dominates the activity here, and the go cue seems to dominate the activity here. So you see, it seems that this go cue activity, this trial task related activity, as well as these unimodal reward responses, is something that is dynamically adapted throughout this task. Okay, so now we'll move on to our testing our main hypothesis. Can we see some information in these responses depending on what trial type follows these cues? So here I'm showing one example, climbing fiber, and here the trials have been separated in these perturbed and adapted trials. And here each dot represents one climbing fiber event or one complex spike, if you wish. So here we see that this fiber produced the same generic activation irrespective of the effect that the stimulus had on the behavior. So we define this type of generic activation as a sensory saliency signal. We used information rate to quantify the predictive information about the behavior, so about these two trial categories that is carried by the absence and presence of complex spikes or climbing fiber events. So you can see for this fiber, the information rate was 0 throughout the trial. We observed this second type of fiber, which was in this case informative about this trial category. So here you can see a clear increase, a clear peak in this information rate, and these red time periods indicate a significant difference in the event probability between these two trial types. So we refer to this type as a motor organization fiber. For this particular cell, so this was a bimodal force and go cue cell, we can also see a second peak, an increase in information after the go cue, suggesting that is also informative about the behavior in the go cue triggered activity. The issue here with these bimodal force and go cue responses is that the second peak might actually be a byproduct of the first peak, given the low firing rate on average of these fibers. So it is highly unlikely that in the same trial, both the force and the go cue will activate this fiber. So to test whether these go cue responsive fibers can be classified in these sensory saliency and motor organization categories, we did not analyze these types of responses. We only looked at the adapted trials so that before the go cue, there is no or only minimal difference in behavior. So here's an example of, again, one generic go cue response that did not differentiate, or there was no information in terms of these two trial categories. So when the auditory tone cued the movement, as opposed to when the tone did not cue a movement. And here's an example of a motor organization fiber. So we have a significant information in the probability of occurrence between these two different trial categories. So we also identified these two types of fibers also for trial that had different kinematics, in this case, fast versus slow movements. And also not showing here, we did also the same analysis for short versus long latency movements, or short versus long reaction times. And the same was also observed for reward-related activity. We're comparing these trials with different latency. So now, if you look at the whole population of image fibers, we see that the majority, 64% of the force-activated ones, are informative of behavior of these two trial categories. One interesting observation is that this information is related to the functional complexity of these cells. So whether the neuron is unimodal versus multimodal. So for example, the percentage of the informative fiber increases as they become more complex. So we can say that we can see here that 78% of the trimodal fibers were informative compared to 41% of the unimodal ones, so almost twice as many. And individual fibers were also statistically more informative. They also had higher information rate values, as can be seen here. For the GOQ related responses, so the percentage of these informative fibers was lower, 17%. So this means that even if fewer climbing fibers were activated in these expert mice, but this force stimulus and more by this GOQ stimulus, these are actually more informative about the ensuing behavior than these GOQ-related responses. So both in higher numbers and also in information rate. So this information, this peak, is significantly larger than this peak here. We also see that for the GOQ activation, that the multimodal fibers are actually more informative compared to the unimodal ones, both in higher fractions of cells and also in higher information rate values per individual cell. For the reward responses, a small fraction could be informative about leak latency, but again, there's much less variability in this behavior that follows rewards. Okay, and these informative force fibers were also topographically more specific to lobule 5 and simplex, whereas these ones were uniformly present in all three lobules. We also found that this type of information can be encoded in the size of the climbing fiber burst, or in our case, the size of these calcium events, which is a proxy for the number of spikes in the burst. So here we compared the size of calcium events that are evoked by the stimulus versus spontaneous activations during baseline. So this is quantified here along the y-axis. So this has already been shown before that evoked events are larger than spontaneous events. But here on the x-axis, we also see that the force-activated climbing fibers produce more spikes for larger events, or perturbed compared to adapted trials when they are responding to these sensory events. We also see the same for the GOQ activation. So evoked events are larger than spontaneous events, but also there's more significant cells in this quadrant here, meaning that cued movements produce stronger bursts in these climbing fibers than non-cued movements. And in the case of the reward responses, there was only a difference between the evoked and spontaneous events and not between evoked events for short versus long latency licks. Okay, and finally, as I highlighted initially, we also analyzed whether climbing fiber activity is modulated in these two periods. So when mice just hold still and wait for the trial to start or wait for the force onset, or where mice hold still and wait for the reward to be delivered. So we call these two different behavioral contexts in the absence of any overt stimulus and behavior. So we compared activity in these two waiting periods in the last 500 milliseconds of both periods. We find cells such as this one, for example. So this was a reward-activated climbing fiber that clearly decreased its activity in the wait for reward compared to the wait for force period. And we also find this type of climbing fiber. So this was a GOQ activated fiber. This was imaged in the same section. So simultaneously, these are two neighboring fibers in the same field of view. And this one increased the probability of having climbing fiber events in the wait for reward compared to this baseline wait for force period. And we observed quite a considerable number of fibers that exhibit these type of modulations by these two behavioral contexts. And when I say a lot of fibers, I mean more than 1/3. So these ones here in cyan with the positive values are like this one that increase their probability while waiting for reward. And these with the negative values are like this one that decrease their probability when they wait for reward. Okay, so interestingly, when we look at the classes of these climbing fibers that have these contextual modulations, we most often see this in fibers that respond to reward. So you can see here, as shown with these arrows, as the fraction of informative cell increases, they become more reward specific. And we don't see any topographic specificity of where these informative fibers are located in these three lobuli. Okay, so I will finish with one last experiment and analysis that Irina did. So this is more of an outlook for future experiments. So she also compared the climbing fiber activity to the postsynaptic Purkinje cell dendritic signals. So we know that most of our knowledge about these olivocerebellar system comes from recording complex spikes or imaging dendritic signals in calcium, in Purkinje cells. But we know from the literature that even though, well, this is a highly reliable synapse, one of the most reliable synapses in the nervous system, there are many factors that can modulate the postsynaptic activity in the Purkinje cell, some of which are outlined here. So we ask whether this postsynaptic climbing fiber responses to the in the Purkinje cells can at least partially be modulated independently of the presynaptic climbing fiber signal. So to test this, as previously, Irina transfected the inferior olive with GCAMP, and then also in these mice expressed RCAMP, a red calcium indicator, in the Purkinje cell, very sparsely, with a Cree-dependent strategy. And then, as shown here, she performed simultaneous dual-color imaging of the presynaptic climbing fiber axonal terminals, as previously, and also the postsynaptic Purkinje cell dendrites. Now, the success of this experiment was rather low because here many things have to work out simultaneously, but we could identify, as shown here, I hope this plays well online, we could identify several Purkinje cell climbing fiber pairs that could be imaged simultaneously. So here's an example of two pairs, so the axonal terminals of the climbing fiber and the dendrite of the Purkinje cell in pair 1 and in pair 2. And you can see here in the extracted calcium traces that these are highly correlated signal. This is the synaptic transmission is as expected highly reliable. We can also see this here in this example trace. We essentially observe 100% transmission reliability. We did, however, observe on very few occasions small climbing fiber events that did not have a concurrent Purkinje cell activation, but this was extremely rare. Instead, what we analyzed more closely is the relation between the sizes or the amplitudes of these synchronous events in these pairs. So you can, for example, already see here in this example that if we take these two events, the first and the last one, they have similar size in the climbing fiber signal, but they have very different sizes in the Purkinje cell dendrites. So to analyze this, we did a very straightforward analysis. So reasoning was as follows. So if these pairs can be modeled as an idealized single input, single output linear system, that any difference between these standardized event sizes should only be due to noise. So in that case, the distribution of the errors or of the residuals between the standard scores, which can also be plotted here as the distance to this unity line, which is directly proportional to this error, this should have a zero mean. And we see for these pairs that in this example, we see that the mean of the error distribution is significantly different from zero here, suggesting some nonlinearity or some residual contribution from non-climbing fiber signals to this Purkinje cell. And this was actually observed in the majority of the 36 pairs that we could image and analyze in this manner. And just for comparison, we also did the identical analysis, but between axonal segments of the same climbing fiber, and observed this non-zero mean in a minority of the pairs, which was significantly lower than this ratio here. So again, this nonlinearity suggests that Purkinje cell dendritic signals has been proposed before, are regulated in vivo by factors other than just the climbing fiber input. And so in the context of our experiments, this means that the information that's encoded by event probability is reliably transmitted at the synapse, but the information that we observe in the size of the climbing fiber bursts might be partially transformed or modulated postsynaptically. Okay, so I will just summarize the main findings from Irina's experiment. So we find, or well, rather we affirm, that climbing fibers can be versatile signal encoders. They have the property of mixed selectivity, which is typically observed in cortical areas that are important for flexible behavior, like the prefrontal cortex. So the Oliver-Sabero system also seems to have this necessary flexibility to organize different forms of behavior. The climbing fibers or the climbing fiber activation can be informative about behavior both in their frequency and also in their sizes of events, in the burst sizes. And the amount of encoded information varies depending on the nature of the sensory event that evokes this activation, but also depends on the functional complexity of the fibers. So multimodal fibers are more likely to encode such information. So what does this mean? This means that climbing fibers with so-called mixed selectivity in one dimension, for example, that are selective for multiple sensory stimuli, are also selective for multiple signals in another dimension, in motor actions, for example. And on the other hand, pure selectivity fibers that represent just one stimulus are also likely to lack flexibility in other dimensions. And finally, we observed that so-called spontaneous climbing fiber activations in the absence of sensory stimuli or movement are not just noise. They also might represent specifics of behavioral context and can also be informative about during these whole or waiting periods where the mouse just waits for the stimulus to occur. Okay, so I would just like to finish here and thank the students in my lab, again, especially Lena here, who spearheaded this whole project, and she recently defended her thesis, and soon she will be on the postdoc market, so please look out for her. Okay, thank you for your attention, and I'm happy to take any questions.
Thank you, Mario, and wonderful. So please just go ahead and raise your hand, and I'll be happy to call on you. You have a question for Mario. One of the more interesting aspects of the work, I thought, was the difference between the naive and the well-trained animals that you showed at the beginning. In particular, I thought the response encoding of reward, the fact that it changed. Would you like to tell us a little bit more about that and maybe give us your intuition about what's happening with regard to the reward response?
Yeah, so again, this is very new behavior. I mean, literally, Irina produced this figure yesterday. So, so the reward is there. I mean, these reward responses are there. They're very prominent in naive mice as well as in expert mice. But really what's striking is that the reward-only cells are completely absent in these naive mice. And they're only present as in these bimodal or trimodal responses. So what might be going on here is that this is a very generic signal initially. So these climbing fibers would respond generically to many different types of stimuli. Then throughout the task, as the reward becomes associated with correctly performing the task, then certain climbing fibers will become specialized for reward. This is a bit the hypothesis that we have at the moment.
Yeah, there's a question in my lab. Please go ahead.
Yeah, we were actually curious about if you could expand a little bit more about the activation frequency and burst size relating to the behavioral outcome. We were curious if you thought that might relate to, so I'm not super familiar with the dynamics of calcium imaging in depth, but I was curious if you thought that might relate to the number of spikelets coming out of the climbing fiber at a given point.
Yes, I mean, these are the presynaptic climbing fiber inputs, right? So, and I believe, yes, that these are related to the number of spikelets in complex spikes. That's possible. Yeah. But again, these are really two different signals. We were looking; these are more a proxy for the number of bursts in these climbing fibers. And I believe there is a relation. If somebody correct me if I'm wrong, but there might be a relation between the number of spikes in this presynaptic burst and the number of spikelets that you might observe in the somatic complex spikes.
Great, thank you.
Beverly, please go ahead.
Yeah, there are some papers showing that there's a correlation between the number of bursts in the presynaptic fiber and the postsynaptic response. The effect is probably going to be quite strong in the dendrites, though. So I would expect more of a strong correlation between the calcium in individual dendritic branches and the presynaptic signal. But of course, this will also be, what you measure will also be controlled a bit by your calcium indicator and the acquisition rate that you can image at. So I'd be cautious with that. But I actually wanted to ask a question. I didn't quite understand the task itself completely, I think. Did you ever, so you were doing all these experiments in expert animals, mostly, I think. Did you change the amount of force that you applied to the lever in animals that are expert and have a look to see what happened to the complex spikes then?
Yes, we did. So I think the goal here was really to try to have a lot of trials in both of these categories, so perturbed versus adapted trials. And so, of course, mice progressively were better at counteracting these forces. So Irina adjusted on a session-by-session basis the amount of the force that she applied. So she progressively increased it so that we always had these two different trial categories, perturbed and adapted.
I see. And so then did you see that the activity was modulated depending on the amount by which you changed the force or the rate at which you changed it or anything like that?
I don't know. I don't have the answer to these questions. I don't know if she looked at that. This is something that we can look at. I mean, we know exactly the amount of force that we applied throughout the sessions, but we haven't looked at this specifically.
Thank you, Beverly. Sasha, you have your hand up.
Yes, I agree with Reza that this changing of the activation of what activates the complex spikes with learning is really interesting. And so my question is sort of related to that and also related to the anatomy. I'm just trying to understand, I think you said something about the complex spikes in different lobules were driven by different types of stimuli, but so the anatomy is such that a given inferior olive neuron will project to multiple lobules the same zone. Presumably you're in the C1 or whatever zone, the interpositus projecting region. But so putting that all together, the question is, are you in, can you tell whether you're in zebra positive versus negative versus mixed zones? And do you think that the relative increase in the activation of the reward signal is because the strength of the MDJ presumed cortical inputs to the olive is increasing? And what's the story with the same sort of olive neurons across lobules? Sorry, take any of that and
Okay. So I'll start maybe with the easy one. So yeah, I mean these are fantastic questions. So the zebra positive versus zebra negative, of course, we didn't do this analysis. We didn't do the identification. I mean, this would again really be a great experiment to do, whether this plays any role in these different categories that we observe. And in terms of projections, I mean, yes, I mean the olive broadly projects to these all different lobules, and I mean we didn't see exclusive segregation of force versus GOQ versus reward in different lobules. We saw responses in all three lobules, but there was clearly a preference for lobule 5 to be responsive to this force stimulus, and lobule simplex to the reward stimulus, which is in agreement, I think, with previous observations as well.
Okay, but is that because you're recording in a or your wonderful student was recording in a visualizing, I should say, in a strip that in which the anatomy is such that you're more likely to be in a zebrin negative zone in the simplex and a zebrin positive zone in the actually I have the answer to this in the anatomy that we've done, but yeah, anyhow, wondering about that, right? So that is, lobules are not uniform structures. Their Purkinje cells and their inferior olive inputs vary as a function of the proximal distal location, right? That is or medial lateral or whatever, depending on how you're oriented. Does that make sense? And so one possibility, maybe this is overkill here, but one possibility is that just because the anatomy of the weird kind of anatomy where the Purkinje cells are folded and stuff like that, or the cortex is folded. Yeah, anyhow, that's you get it.
Yeah, I mean, so I mean, one thing maybe worth mentioning here is that we don't have so these are not a transgenic mice where we uniformly express this calcium indicating Purkinje cells like I think Dimitar is doing in the Horser lab. So we don't have a uniform expression across the whole lobule in these climbing fibers. So we inject in the inferior olive and we really image the regions where we see most of these axonal terminals. And perhaps with a more uniform sampling of the cerebellum, then we can go into these questions, how different divisions of different lobule encode information.
Got it. Cool paradigm, though.
Thank you. Thank you,
Thank you.
Sasha. Dimitar.
Hi, thanks. I really enjoyed your talk, and I really like your preprint. I think it's a really nice paper. I was going to say, in part to answer Sasha's question, medial simplex has like really closely alternating zebrin positive and negative bands if you look at the staining, and so you're probably recording from a combination of the two, as we usually do as well. The question I had was about the last thing you showed about the variability in the transmission between the olivary neurons and the climbing fiber terminals and the Purkinje cell dendrites. I wonder if you ever compared across, I guess like you're clustering and then correlating across multiple presynaptic terminals that you think are belonging to the same climbing fiber, right? I wonder if you ever did a similar analysis to look at the variability amongst those climbing fiber terminals and compared that to the Purkinje cell dendritic events, which would be the kind of nice internal control for the spatial differences across your Purkinje cell dendrites that you say are more uniform, right?
So I mean, the control experiment that I showed, we just took wherever we see a visible climbing fiber that could be actually divided into segments. So we did look at that. But we did look also and there, I mean, we don't see as much.
Totally. But but did you ever do a comparable thing looking at different terminals that you think belong to the same climbing fiber?
Yes, so Irina, I think, spent a lot of time looking at that, and we do see variability, which is quite surprising. So it seems that axonal terminals belonging to the same fiber do not correlate perfectly. There is some variability there between terminals. And cool. I think, I mean, recently we've seen a talk that, if I'm not mistaken, so there's similar variability in other brain areas if you just follow the axonal terminals of the same neuron. It's not always uniform. There's quite some variability there. I don't want to say that, but I think these were striatal projections from the cortex, and we also find a lot of variability in the axons, in the axonal terminals on the same branches.
Cool. Yeah. And I guess it's still early days to speculate on the mechanism for this.
Oh yeah.
Yeah. I guess it could be like there's old papers showing that neuromodulators can activate or can change release probability from.
Yeah, so there's strong aminergic modulation of these synapses.
Yeah,
so,
exactly. Exactly. That's really cool. Thanks.
What a wonderful, absolutely wonderful discussion. Are there any other questions for Mario? I was gonna maybe mention something and see what your thoughts are regarding this change in encoding of reward, and depending on the expertise of the animal, and in effect asking questions regarding whether we can understand these changes in terms of how the rest of the brain evaluates that sensory event, or even the motor event, meaning that the evaluation of the reward as it's changing for the animal and being reported potentially to the inferior olive, that potentially is changing. And that then the response that the olivary neurons have and how they transmit that information is a reflection of a change in the value-based evaluation of events, for the essentially how that reporting might be changing for the cerebellum. I don't know if that makes any context sense to you, Mario.
Yeah, it does. I mean, so yeah, I think that's a great question. So the task that we have is, I mean, quite simple, right? It's quite low-dimensional behavior. I mean, this type of questions that should be investigated, I think, with a task that's typically done for the dopaminergic system, where you make the reward uncertain, and then look at these anticipatory signals and how they modulate in terms of the uncertainty of the reward, and whether there's some learning there over time.
Yeah.
But I think that we require a much better task, or a specific task design to address this question.
Wonderful. Well, thank you, everybody. It's wonderful to see you. I hope you have a wonderful rest of your summer, and many of you I hope we'll see in Switzerland at the Gordon Conference. Until then, take care. Bye-bye.
Thank you. Bye-bye.